Good morning, ma'am.
Good morning, ma'am. Good morning, students. Can you hear me, children?
Yes, good morning, ma'am.
Yes, ma'am. Yes, ma'am.
Good, it was conveyed. I was thinking, I don't know how many of you will be able to join. So, beta, if somebody comes in waiting, let me know. Huh? I'm starting my class. I'm in a hurry today. Today, we are going to study something very important in biotechnology, that is vectors. Okay, beta? Anybody knows what is a vector? Malaria में पढ़ा होगा, when you have done malaria disease when you were small. Which organism acts as a vector in malaria? Tell me, beta, quickly. What is the causative organism of malaria? P से start होता है. Name the causative organism of malaria. It is Plasmodium. And what is the vector? Normal language में नहीं बोलते हो कि mosquito bite से malaria होता है. Name the vector in malaria.
Female Anopheles.
Female.
So what does female Anopheles actually do? It carries the pathogen into the host body. यही करता है vector.
Yes,
So something like this, we are going to study about vectors in biotechnology today. They are very important. I have put one arrow on the right side and one arrow on the left side to tell you that what are vectors actually. See, beta, they are nucleic acids. If I just explain it to you, what are vectors? They are nucleic acids. They could be DNA or they could be RNA, anything. But in 99% cases, because genetic material is DNA, so vectors are DNA. But that does not mean that only DNA acts as vectors. In certain organisms, RNA also acts like a vector. Chalo. What does a vector do? Dekho, main property of this vector. It's mainly a DNA molecule which can carry a foreign DNA segment. Foreign DNA segment pata hai na? Foreign DNA segment means
Yes,
the
ma'am.
DNA. Everybody knows what is foreign DNA. जो चीज अपनी नहीं है वो foreign DNA. Supposing I want to incorporate a foreign DNA, I want to put my desired gene. याद है? Everybody remembers the word desired gene?
Yes, ma'am. Gene of interest.
Supposing I want to insert my desired gene in an organism, किसी भी organism में, I want to insert the desired gene. So I need the help of these. I cannot do it directly. I need the help of these. They are vectors. So what is vector? It is DNA or RNA. It could be any molecule. It could be DNA molecule. It could be RNA molecule. So mainly vectors, they are DNA molecules which can carry a foreign DNA segment. So पहला काम vector क्या करेगा बेटा? It will carry that foreign DNA segment into the host. Desired gene जो आपकी है, it will carry the desired gene into the vector. Sorry, into the host. Host में जाने के बाद it will do this work. It will replicate inside the host cell. It will start replication. आपने तो एक foreign gene insert करी, and after it enters the host 1222224, and the number of genes will start replicating. Got everybody? What are the things which a vector is going to do? What is a vector? It is a DNA or an RNA molecule which can carry a foreign DNA segment and it can replicate inside the host cell. बेटा फायदा क्या होगा? अगर ये रेप्लिकेट करेगा, आपका डीएनए रेप्लिकेट करेगा, तो डीएनए के साथ हमने क्या इंसर्ट करा है इसमें?
Desired gene.
Desired gene. So the desired genes will also replicate. You want that character to be there in the organism. That is your desired gene, desired character. So automatically what happens is, if this will replicate, then the desired gene will also replicate. Chalo, let us understand the first three lines. Vectors, right side, it's mainly a right. वैसे तो if you say vectors, they're nucleic acids. It could be DNA or it could be RNA, which we utilize to transfer a desired gene. Now, what are the main things? What is vector? सिर्फ समझने के लिए, mainly it's a DNA molecule which can carry a foreign DNA segment and then it can replicate inside the host cell. Supposing I want कि एक क्लास में से 50 कार्ड्स बने, तो मैं क्या करूंगी? I will just tell one child, I'll pick up one child, this vector, and इसको एक कार्ड दे देंगे और जाके बोलेंगे जाके क्लास में बेटा ऐसे बहुत सारे बनाओ. So वो एक सैंपल जो कार्ड था वो कौन लेके जा रहा था? This child, that is this vector. क्लास में जाने के बाद, class is your host. After entering the class, what did the vector do? उसने सैंपल दिखाया and all the students started making so many cards of the same kind. वो process हो गया आपका replication. What would that process be? Replication. So vector was carrying what? हमने देखो बेटा, हमने एक स्टूडेंट को यूज किया कार्ड लेके भेजने के लिए. हम यहाँ पे कार्ड नहीं भेज रहे हैं. What are we sending? We are sending a desired gene, and we are sending it through whom? We are sending it through a vector. This vector, it will carry the desired gene into the host cell, and there your gene will start replicating. बेटा vector का काम समझ में आ गया?
Yes, ma'am.
Yes, ma'am.
Everyone understood what is a vector too? Very simple. If I say in one line, it will carry a foreign gene or foreign DNA segment, and then after it reaches the host cell, it will start replicating. But हम किसी भी बच्चे को तो सेलेक्ट नहीं कर सकते हैं ना वेक्टर के लिए. We need some child जो अंदर जाके मोटिवेट करे बच्चों को. There may be students जिनको मैं ये काम दूं, they may enter into a class and they may not take interest in this. So all the class, will they be involved in card making? They will not be involved in card making. That means replication process नहीं होगा. And if replication process is not taking place, then that vector is useless. Got my point, beta? Yes,
Yes,
or
ma'am.
no?
Yes,
So,
ma'am.
Yes,
every
ma'am.
organism,
Yes, ma'am.
good. Every organism cannot be considered as a vector. किसी को भी उठा के आप एक representative student नहीं बना सकते हो. Like I just told you, we cannot have every organism acting like a vector. There are some characteristics for an organism to act as a vector. So, किसको vector select करेंगे? There are some qualities that that particular organism should have so that it can act like a vector. See on the screen the first quality. Please read out loudly what is the first quality.
Able to replicate.
Able to replicate.
It should be able to replicate. Now, able to replicate का मतलब it should have this ORI site, because ORI site is the site जहाँ पे replication start होती है. Is it okay? So obviously, if we wanted to replicate, तो उसके अंदर which site should be present? ORI site should be present. And why this site should be present? Because this is the site where what starts is your replication starts. This is the site where replication starts. Beta, what is the first quality of a vector? It should be able to replicate. And to replicate, it should have this site. Why it should have this site? Because it is this site जहाँ पे replication start होगा. Is it fine? Now, the question arises why? Why do we want to replicate? Okay? Why do we want to replicate this DNA segment? ये जो छोटा सा DNA segment हमने भेजा वेक्टर के फॉर्म में, इसका replication क्यों कराना है? Why do we want it to replicate?
Because it carries the desired gene.
Just it carries the desired gene. अगर वो replicate करेगा तो desired gene की भी बहुत सारी copies बन जाएंगी, which we want. That is the main reason that we want it to replicate. So the first and the most important characteristic of a vector is that it should be able to replicate. After entering the host, if it is not replicating, तो आपकी single gene रहेगी. Single gene will not be able to give you the desired effect जो आप चाह रहे हो. So it is useless. It is no point using such a vector. It should be able to replicate after entering. into the host, and when it will replicate, automatically desired gene की बहुत सारी copies बनेंगी. So the first quality which a vector should have is it should be able to replicate. And for replication it should have which side, children?
Ori side.
Ori side.
Each side.
Because this is the side जहाँ पे क्या चीज start होती है?
Replication.
Replication.
We said कि
Replication.
वेक्टर की सबसे मेन क्वालिटी रेप्लिकेट करना, तो रेप्लिकेट तो तभी होएगा व्हेन वी हैव दिस साइट. एंड क्यों रेप्लिकेट कर रहे हैं? व्हाई आर वी रेप्लिकेटिंग? बिकॉज दिस कैरीज योर डिजायर्ड जीन. लेट मी एक्सप्लेन इट टू यू विद एन एग्जांपल. सपोज वी इंट्रोड्यूस इंसुलिन प्रोड्यूसिंग जीन इनटू ई कोलाई. दैट डे आई हैड डन दिस इंसुलिन वाला. सबसे पहले दिन जब हमने बायोटेक्नोलॉजी स्टार्ट किया था, वी सेड we have what is a desired gene over here? Insulin producing gene. Okay, which is the desired gene, children?
Insulin.
Insulin producing gene. And आपका host ये है. What is your host?
E. coli.
E.
E. coli.
I said कि हम desired gene को host में डालते हैं with the help of a vector. So here we will use a vector. Which is the vector we are going to use?
Plasmid.
Plasmid.
If you remember that circular वाला thing I had made, जिसको cut करके फिर हमने linear बना दिया था. Okay.
Yes, ma'am.
That is what we call. That is what we call as. What is that called as? That is your vector over here. अब हमने example समझने. We are inserting our desired gene, children. Which is our desired gene? जल्दी जल्दी बताइए quickly.
Insulin producing gene.
Insulin producing gene. And we want many genes जो insulin produce कर सके ताकि हम insulin extract कर सके E. coli से. So the main thing is कि this gene should undergo replication. So we have introduced insulin producing gene. Host है आपका E. coli. Vector हमने use किया plasmid. Now what happens is this plasmid enters, replication process starts. This plasmid will also start replicating. As soon as it enters the host cell, plasmid will start what? It will start the replication. And I told you before only that for replication, what will it need? What site is needed?
Ori
Ori.
side.
Ori side.
So, yes. If this is replicating, that means आपका ये plasmid replicate कर रहा है, तो automatically what is a replicating, children?
The
See
gene
this,
which
what is?
Desired gene.
Yes. The desired gene will also start replicating. I'm reading it out for the last time, the first point. The first important characteristic for a vector, for an organism to act like a vector is what? Tell me quickly, beta.
Able
It should be able to replicate.
Yes, it should be able to replicate. And to replicate, it should have its site.
Ori side.
Ori side.
Ori side.
And why does it need to replicate?
To get
Ma'am.
desired gene.
Because it carries desired gene.
Ma'am, it's carrying the desired gene.
It carries the desired gene. And we have taken an example to understand कि vector कैसे काम करता है. What is the desired gene which I have discussed in the example just now?
Insulin.
Insulin.
बेटा, अगर आप एक
Insulin.
जीन को इंट्रोड्यूस करोगे, if you have only one insulin producing gene, तो इतना इंसुलिन नहीं मिलेगा that you can extract it from the host. So we need to have more number of such genes. Insulin producing gene was introduced in which host, beta?
E. coli.
E. coli.
Okay. Which is the vector that I'm using?
Plasmid.
Plasmid.
So I told you important property vector की होनी चाहिए that it should replicate. So this plasmid will go and start replication. So once it replicates, what site it should have? This plasmid should have this site. तभी वो replicate कर पाएगा. What is the site?
Ori side.
Ori site.
Ori
Because
site.
it is from here that it will start replication. And जब ये replicate करेगा, when this replicates, your desired gene will also start replicating. Any confusion in the first quality?
Ma'am, which topic are
No,
you studying?
ma'am.
No. We are moving on to the second quality of vectors, that is ROPs. Okay, beta. Now understand what is ROP. This is the
Molecular
protein
basic
which is needed for replication. Is it okay, beta? Although we are using a vector for replication of the desired gene, but there are certain other things which should also be present. So what is the second main quality which a vector should have? एक ORI site I have already told you. Yes, I already told you कि ORI site होनी चाहिए. एक चीज और which is essential, that is ROP. Understand this ROP. This is the protein for replication. अब try and understand ये protein कैसे help करेगा replication में. If you remember, we had done the working of DNA polymerase. DNA polymerase आके attach करता है अपने आप को at a particular site या ORI site. You remember?
Yes, ma'am.
Yes,
With
ma'am.
transcription and translation, we said na DNA polymerase will come and attach itself to a particular site, ORI site. फिर वो धीरे धीरे आगे
Yes,
बढ़ता
ma'am.
है, and sometimes एक blockage आ जाता है. It is not able to, then transcription stop होता है. We had done that, if you remember. Now, this enzyme of yours, DNA polymerase, ये identify कैसे करता है कि which is that site जहाँ पे जाके मुझे attach होना है. Okay? Here, DNA polymerase will be helped by this protein. The name of these proteins are ROP proteins. Okay? Replication proteins basically we call them. So, what is it, beta? How they are going to help? अब समझो इसको. This protein binds with the ORI site. This protein, what will it do? It will go and it will bind with which site?
ORI site.
ORI site.
अब DNA polymerase has to come and attach itself to which site? ORI site. So that अब DNA polymerase को help करेगा. ये कैसे help करेगा? DNA polymerase will recognize these proteins. जहाँ पे भी ये protein है, that means वो कौन सी site है?
ORI site.
ORI site.
ORI
So
site.
ORI site.
first your protein will bind at the ORI site. Then DNA polymerase will identify the ORI site because of this protein. जैसे क्लास के बाहर एक नेम वो लगा रहता है 5th A, 6th A, 9th A. What does the teacher do? Teacher is a DNA polymerase. Teacher has to enter a class. She is new in the school and teacher is a DNA polymerase. तो वो क्या करेंगी? वो सबसे पहले वो देखेंगी कि क्लास का नाम क्या लिखा है. Once she sees okay, 11th भी लिखा है, then she enters. Exactly वही चीज यहाँ पे होगी. Your teacher is your DNA polymerase over here. And the class board जो बाहर sign board लगा है, that is your ROP protein. So what will the ROP protein do? ROP will go and bind itself to which site, children?
ORI site.
ORI site.
ORI site.
So जब DNA polymerase
ORI
आएगा,
site.
first protein will go and bind itself at ORI site. Then your DNA polymerase will come. It will identify the ORI site because of the presence of what on those sites?
Protein.
ROP protein.
Protein.
Because they don't have
protein.
any site. So DNA polymerase has to attach itself only to the ORI site. And the identification is possible only because of the presence of these proteins. So which was the first main quality of vectors which I discussed with you before this, the first one?
Able to
It should be
replicate.
able to replicate.
Able to replicate.
Replicate. Excellent. And second is the presence of proteins. What are the proteins called?
ROP.
Replicate.
Protein
ROP.
for replication.
A third important quality of vectors we are doing is the presence of at least one selectable marker. In the bracket, I have written any one. जरूरी नहीं है कि दो चाहिए हो, दो तरह के होते हैं, but we need any one of them. At least one selectable marker has to be there. अब इसका function समझते हैं पहले. I think you people, when you study notes बनाते हो, you use markers to markers, highlighters in your books. You do that or you don't do this?
So, ma'am,
Why do you use highlighters or markers in your notes or books? You don't
And to
use?
highlight important points.
हाँ, so that जैसे ही आप पेज खोलो, आपको पूरा पेज नहीं पढ़ना पड़ेगा. You have to just see, okay, these are important. Immediately,
Yes.
you will be able to identify what is important. That is the work of a marker. Yes or no?
Yes, ma'am.
Yes,
ma'am.
We are going to understand what are markers in biotechnology. Okay, there are many genes which can be used as selectable markers, but remember that these markers should have at least, I'm talking about the vectors. Vectors should have at least one. Any one selectable marker should be there. Selectable marker क्या करेगा बेटा? हमने क्या करा था? We took a desired gene, children, and we inserted it into a host with the help of what? What did we use
Please
to
repeat.
insert? We took a desired gene and we are inserting it into the host cell with the help of what? I have just
Vector.
started with the help of a vector. With the help of a vector. Now the vector has
Yes, ma'am.
started replicating. That means your gene has also started
Replicating.
Replicating.
But how do I know? How will I check that my gene has entered the host or not? कि vector ने कुछ गलती तो नहीं कर दी है? whether the vector has carried my gene or not. Is my gene replicating or it is not replicating? कहाँ पे gene present है? I need to see, I need to identify my gene or not.
Yes, ma'am.
Yes,
Out
ma'am.
of so many genes, I want to just identify my gene, the one which I inserted, yani gene of interest, whether it has entered the host, if it has entered to replication हो गया कि नहीं हो गया, how many copies are being made of the gene, and कहाँ पे the copies are present. So for that, I use special genes which are selectable markers. Understood the work of a marker in biotechnology?
Yes, ma'am.
It will identify your foreign gene after it has entered the host. It will be able to identify those. So let us study about markers. I said the main third quality of a vector is it should have at least one marker. There are two types of markers in biotechnology, children. So in दोनों में से at least one should be present. Let me tell you now. Gene which can be used as a selectable marker. You cannot pick up any gene कि चलो इसको selectable marker बना लेते हैं. We cannot take it any gene as a selectable marker. There are two types of genes which can be used as selectable markers. The number one are antibiotic resistance genes. Which is the first kind of gene which can be used as a selectable marker?
Antibiotic resistance.
Antibiotic resistant gene. कैसे check करेंगे बेटा that this gene is present? Supposing I have taken resistance against any antibiotic, tetracycline or any supposing. ठीक है? I have this antibiotic resistant gene. So this will act like my selectable marker. The second one which can act like a selectable marker is the enzyme producing gene. Which gene?
Enzyme
Enzyme producing gene. Here I give you an example of antibiotic resistant gene. It could be any antibiotic, ampicillin, tetracycline, any antibiotic resistant gene you can use. That means resistance against any antibiotic. The second kind of a gene which can be used as a marker is enzyme producing gene. Let us do this example. Normally, in this case, बहुत कम case, बेटा ये तो बहुत सारी होती हैं. Antibiotic resistant genes are many. But we talk about enzyme producing genes, they are very few. Normally in biotechnology, we use this gene. See the name of this gene. What is the name of this gene? Read out.
Lecture.
Yes, the name of the gene is LAC-Z gene. Okay. The name of the gene is LAC-Z gene. This will go and synthesize beta galactosidase. Which enzyme it synthesizes? Which enzyme
beta
it produces?
galactosidase.
Beta
Beta
galactosidase. So these markers, what is the function of the markers? They help to identify if the desired gene is introduced into the host or not. चलो check करते हैं कि कैसे. Supposing I have introduced this gene. Which gene? Antibiotic resistance gene. And now I expose my host towards tetracycline. Then I will check that has my host developed immunity or resistance to this antibiotic or not. Agar yes, that means my desired gene is already introduced into the host. Similarly, agar ये enzyme produce होना शुरू हो गया, कौन सा वाला enzyme I talked about just now?
Beta
Beta
galactosidase.
galactosidase.
That means this gene of mine is already inserted. Which gene I had inserted over here?
Lecture.
LAC-Z.
So very quickly, which is the third thing a vector? The first thing a vector should have was
Able to replicate.
Able to replicate.
able to replicate. That means it should have which sites?
Ovocyte.
Oocyte.
The second thing that we did was each vector
Protein
should
for
have
replication.
Protein for
protein.
replication.
What is the name of the protein?
ROP.
ROP.
ROP goes and binds itself to which site?
Oocyte.
Which enzyme comes and identifies that protein?
Ma'am, what?
Which enzyme comes and binds?
DNA polymerase.
Very good. DNA polymerase. Which is the third thing which every vector should have? At least one it should have. At least one it should have. What is
One
the
marker.
Selectable marker.
बेटा इसका ध्यान रखना. At least one, any one. Both of them are not needed. So there are two. We just need any one of them. The first one was your antibiotic resistance gene, and The second one is what? Your second one is your enzyme producing gene. Just now we talked about maximum cases, beta. The normal gene which is used in enzyme producing gene is which gene?
Lactic.
Laxate.
Lac gene synthesizes an enzyme. What is the name of the enzyme?
Beta-galactosidase.
Beta
Beta-galactosidase.
galactosidase.
So these markers will help you to identify if your desired gene is introduced into the host or not. And the fourth thing which we are going to talk about that vector should have is a restriction site. Okay, it is what we call as restriction site. We know that we should be able to cut the vector by endonuclease. We can cut the vector by endonuclease. That day I had cut one vector by endonuclease, a circular vector. What was the name of the vector?
Plasmid.
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 Very good. Circular vector liya tha, we had taken plasmid and we had cut it with molecular scissors. What was the name of that enzyme with which we had cut?
Endonuclease.
Endonuclease.
That means ऐसी कोई site होनी चाहिए where endonuclease can cut that particular vector. That site is what we call as the restriction site. Okay, so what do you mean by restriction site? We should be able to cut the vector by endonuclease. At least one site to ऐसी होनी चाहिए बेटा. Otherwise, what will happen? Your endonuclease will not be able to cut the vector. And if you're not able to cut the vector, then it is useless. आपका कुछ insert नहीं होगी foreign gene that you want to insert. It is not possible to insert. So we should have one restriction site so that we are able to cut the vector by endonuclease. Don't get confused. Restriction site का मतलब क्या होता है? I put an arrow. Later I realized after writing that maybe you don't understand. So restriction site is the place where endonuclease cuts. What is restriction site, children?
Place where endonuclease.
Place where endonuclease comes.
Endonuclease is cutting. So we should have one restriction site होनी ही चाहिए. But remember, we should have only one restriction site normally in vectors. अब देखो, I have tried to draw a vector. If you can see the circular vector over here on the screen, and I have made so many restriction sites. Can you see how many restriction sites are there? 1, 2, 3, 4. Count karo. 5, 6, 7. 7 restriction sites. Up, if I have 7, though abhi I'll be taking up an example जहाँ पे हम artificially बनाएंगे तो 8 restrictions. अच्छा, I'll explain it to you. अभी confused मत हो. See, this is a circular vector. These are restriction sites. If many restriction sites are there, then the vector can be cut into many small pieces. Clear? Over here, it can be cut into many small pieces. So if it is cut into many small pieces, क्या problem आ सकती है? Can
मैम
anybody
वो
think and
डैमेज हो
tell me?
जाएगी. मैम वो ओरी
Ori
साइड
side
डैमेज
damage
हो
हो
जाएगी.
जाएगा.
ओरी साइड डैमेज हो सकती है. हो सकता है मैंने यहाँ पे मार्कर इंट्रोड्यूस करा हो. यहीं
Yes,
पे
ma'am.
एंडोन्यूक्लियस ने कट कर दिया तो my marker will also get destroyed. Okay, and your ori site can also get damaged. Ori site damage हो गई तब तो कुछ होगा ही नहीं. Replication process will not start only. So we prefer that it should have only one restriction site. Though abhi I'll be taking up an example. जो हमने बनाया है the first artificial one clone vector. We have 8 restriction sites in it. I'll be coming to it very soon. So have you understood the fourth thing? Restriction site means the place where endonuclease is cut. Each vector, we should have one restriction site so that your endonuclease is able to cut the vector. So this is the fourth thing which every vector should have. And see over here, it should have one to open and insert the desired gene. Supposing I have one restriction site, I will cut the enzyme will cut from here. And then it becomes like this. You remember that day I did, this is your linear one. Cut करने के बाद वो finger वाला बनवाया था, thumb and finger.
Yes, ma'am.
We had opened it. So if it cuts at one side, it will open, it will become like this. And you can insert what over here? What is this inserted, children? This is your
Gene of
desired.
interest.
Gene of interest.
So it should have only one so that it opens up. And after it opens up means ये circular वाला चीज it opens up and we can introduce the desired gene. Fifth quality which it should have is it should be small in size, because then it is easier to handle small vectors. It becomes difficult to handle big vectors. It is always easier to handle small vectors. Before I move on to a very complicating topic, there is one vector in your syllabus which is in detail you have to do. PBR 322 structure we have to learn. We have to draw. It is something like this. PBR 322 ये वाला structure है. Before I move on to this, I want you to first quickly revise what I see. This is PBR 322. It's there in your syllabus. Only one vector is there in your syllabus. इसको हम detail में पढ़ेंगे अभी. Very quickly, we are going to do vectors. A fast revision of what we have done vectors. See, I said what are vectors? Basically, they are nucleic acids. They could be DNA or they could be small strands of RNA which we utilize to transfer our desired gene. Vectors are mainly DNA molecules which can carry a foreign DNA gene, and then they should go inside and replicate inside the host cell. Which was the first characteristic, quickly, children?
Able to replicate.
Able to replicate.
They should be able to replicate. And they should be able to replicate. For that, they should have which site?
4E site.
Story site.
And why replication is needed? Because they are carrying which gene of yours?
Desired gene.
Desired
Desired.
gene.
The second thing which all vectors should have is
Protein for the plate.
Protein.
protein. What is the name of these proteins?
ROP.
ROP proteins. ROP proteins go and bind themselves to which site?
Ori site.
Story site.
So that which enzyme can identify the ORI site?
DNA polymerase.
DNA polymerase.
Because
DNA
DNA
polymerase.
polymerase आएगा, then replication process start होगा, and main purpose is replication. So we are done with the second characteristic. Third thing which each vector should have is
One
Marker.
one selectable marker. We should have at least one. There are two types of selectable markers generally used in biotechnology. A gene which can be used as a selectable marker is, which is the first kind of a gene which can be used?
antibiotic.
Antibiotic resistance gene.
Anti-resistant genes. And the second one which can be used is
Enzyme producing.
enzyme producing.
Enzyme producing gene.
We have done an example of an enzyme producing gene. The name of the enzyme, LAGZEC. It synthesizes which enzyme?
Galactosidase.
Very good. So these markers help us to identify if your desired gene is introduced into the host or not. And the fourth thing which each vector should have is what site?
Restriction site.
Restriction site.
When I say restriction site, it means it is the place where what happens?
Back
Endonuclease.
cut by endonucleases.
The place where endonuclease cuts. Okay, beta? Now, it should have only one restriction site, preferably, because where if it is cut at many places, then your ORI site may get damaged, your markers may get destroyed. And most important, your gene of interest may also get destroyed. So it is no point that replication process will not start, gene ही नहीं बची है. Next, it should have at least one. एक तो होना ही चाहिए. ठीक है? चलो. It should be small in size because it is easier to handle which vectors?
Small vector.
Small vector.
So whenever this question comes that what are the characteristics of vectors, first tell what are vectors, that they are DNA or RNA molecules which inserted into the host start replicating the desired, start replicating. They carry the gene of interest and the gene of interest also starts replicating. How many characteristics will you mention, beta?
5 characteristics.
Achha. I think I just have two minutes. Let us try to understand about the vector which is there in your syllabus. Structure तो आज नहीं करा पाएंगे, but इतना तो I think I can do. See, the only one vector is there in your syllabus, that is PBR 322. बस इसका नाम कहाँ से आया ये समझ लेते हैं. Okay? Tell me the name of the vector. It's very important. The structure is very important. What is the name of this vector?
PBR 322.
This is E. coli cloning vector. This is actually a cloning vector used with E. coli. Okay, so PBR 322 is E. coli cloning vector. Let us do this very quickly. P stands for plasmid. Okay, beta, this letter, can you see P? It stands for plasmid. BR, these two letters we've taken from the name of the scientists. B over here is Bolivar, and R is your Rodriguez. Okay, अब जल्दी से बताओ, what does P stand for?
Plasmid.
Plasmid.
E and R are the name of the scientist. B is who?
Oliver.
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 And Gulliver nahi, Gulliver Travels wala Gulliver nahi, it is Gulliver. And R is what?
Roderick.
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 And this 322 is the code which is given to this plasmid. There are many plasmids. So codes have been given to plasmids. So this code, this plasmid of E. coli cloning is given a code. What is the number of that code?
322.
322.
Too important. This is very important because this was the first artificial cloning plasmid. This was the first artificially cloned plasmid. What do you mean by artificially cloned?
Man-made.
हमने खुद बनाया मतलब
हमने खुद बनाया, क्लोन कर लिया, उसकी क्लोनिंग कर ली नेचुरल प्लास्मिड की, इसकी हमने क्लोनिंग करी नेचुरल प्लास्मिड की. वी आर टॉकिंग अबाउट ई कोलाई प्लास्मिड. ई कोलाई का क्लोनिंग कर लिया हमने, ई कोलाई प्लास्मिड की क्लोनिंग करी एंड फर्स्ट आर्टिफिशियली हमने उसको बना लिया. वी क्रिएटेड द फर्स्ट आर्टिफिशियल ई कोलाई क्लोनिंग प्लास्मिड एंड व्हाट नेम वाज गिवन टू दिस PBR 322. P बिकॉज हमने प्लास्मिड किया. यस बेटा.
मैम ये 322 मैम ये रोमन न्यूमरल
नहीं, इसको रोमन में नहीं लिखते हैं. रोमन में हम उनको लिखते हैं जब एंडोन्यूक्लियसेस के नाम देते हैं. ये देखो नीचे, ये सी दिस, आई हैव यूज्ड दिस ओवर हियर, सी ओवर हियर, आई स्टार्टेड यूजिंग दिस 1 2 3, ये रोमन में चलेगा. ओके, दैट वाज स्ट्रैंड. दिस इज एंड दिस शोज द सीक्वेंस ऑफ डिस्कवरी. ओके, अभी रुक, ये जो रोमन नंबर्स होते हैं ये सीक्वेंस ऑफ डिस्कवरी. लाइक हिंद 2, हिंद 1, जो पहले डिस्कवर हुआ वो 1, जो बाद में हुआ वो 2. दिस शोज सीक्वेंस. And ये जो है, this is the code which is given to the plasmid. There are many plasmids. So here E. coli की plasmid की बात हो रही है, and the code which is given to E. coli is 322. What is important about this plasmid is that it is the first artificial cloning plasmid. Second thing which is important is, why is this structure so important, beta? There are many restriction sites. अभी थोड़ा सा count करने की कोशिश करो. चलो count करते हैं. This is 1 2 3 4 5 6 7 8 restriction sites. There are 2 antibiotic resistant genes. I said एक भी होगा तो काम चलेगा, but here you have 2. If you can see over here, this is ampicillin resistant gene, this is tetracycline resistant gene, and of course ओरेसाइड तो जरूरी है ही, and ROP भी present है. Okay, this is the structure of the first artificial plasmid cloning plasmid made. What is the name of this cloning plasmid, children? Quickly.
PBR 322.
PBR.
What does P stand? What does P stand? परेशान मत हो, this structure I will make you learn with me in the class only. बिल्कुल परेशान मत हो. P stands for plasmid. B stands for the name of the scientist. Who is the scientist?
Bolivar.
Bolivar.
And R stands for?
Roderick.
This 322 is the code given to the plasmid. This is a very special vector because this is the first artificial cloning plasmid. It has how many restriction sites?
8.
8 restriction sites, and it has 2 antibiotic resistant genes. This is one ampicillin resistant gene, and this is tetracycline gene. It has ORAE, it has ROPs also. So all the 4 things are present, and it is also small in size. If you see, all the 5 characters are there. However, ज़्यादा है, that I'll tell you why. 5 things are already there. We have 5 these 8 sites, 8 restriction sites, 2 antibiotic resistant genes. चलो जल्दी से बताओ. How many restriction sites in PBR 322?
8.
How many antibiotic resistant genes?
2.
Name them. I have a class at 9 o'clock for English.
Ampicillin resistance
Ampicillin resistant gene
gene,
and tetracycline resistant gene.
Yes, tetracycline. Yes. Next class again I have in your class only. I'll be finishing my classes at 9:45 today. इसके बाद immediately about English in your class. Ampicillin resistant gene and tetracycline resistant gene. And does it have an ORI site?
Yes,
Yes, ma'am.
ROP? Does it have an ROP?
yes, ma'am.
Yes, ma'am.
Is it small in size?
Yes, ma'am.
Yes, ma'am.
All the 5 characteristics are fulfilled. I do have restriction site, I have antibiotic resistant gene, I have ORI, I have ROP, and it is also small in size. That is why this is a perfect vector. This is a perfect vector. So we are going to study the structure of this. बहुत सिंपल तरीके से कर सकते हो, बस ये याद रखो there are 8 restriction endonuclease sites in this. Okay, how we are going to function this, we'll start doing this very quickly. चलो अगर टाइम है तो सिर्फ फर्स्ट वाले सेक्शन को समझते हैं. These days इंडिया एंड चाइना में वॉर चल रहा है. Yes. Ampicillin resistant gene and tetracycline resistant gene. The third thing that you will draw in the smaller circle inside is your. Is it clear? When we start drawing, first we will make what circle? We will make a big circle. Inside the big circle, we'll make a small circle. Inside the small circle, we'll write 4 things. What are the 2 resistant genes which you will write, beta?
Ampicillin resistant and tetracycline.
Yes, ampicillin resistant gene, and second is your tetracycline resistant gene. What is the third thing which is written?
Tetracycline.
ROP.
ROP. ROP is the protein needed for
Replication.
Yes, it is the protein which is needed for replication. And what is ORI?
Origin of replication.
Site for replication.
Site.
Yes,
for
it
replication.
stands for origin of replication. Now we'll together make out some way to learn this. Then we'll see कि आपको कितना याद हुआ. These days सबसे पहले ऊपर वाला लेते हैं, this part. Border पे there is a lot of tension
India and
between
China.
India and
India.
India and China.
India and China. So let's talk about India as Hindustan and China. So सबसे top priority for the country this time is Hindustan-China relations. This is how we will try to learn. Who started this dispute first? Hindustan or China?
China.
So C will be given as Roman number 1. And हिंद तो बहुत लास्ट में हम आते हैं, वी डोंट गेट इनटू फाइट्स. सेकंड ऑप्शन हम लेते हैं कि थिंग्स शुड बी सॉर्टेड आउट पीसफुली. अगर वो नहीं होता है तब हमने अपना आर्मी को वहां पे लगाया. सो हिंद विल बी 3. सेकंड ऑप्शन वाज पीसफुल कन्वर्सेशन, जो नहीं काम आया, सो वी हैव टेकन द ऑप्शन एज 3. सो फर्स्ट बॉक्स एवरीबॉडी इज क्लियर. वी आर गोइंग टू टेक एज C and H. C over here is CLA. I'll tell you ये CLA क्या होता है, इको क्या होता है, I'll tell you later, but आपको लर्न नहीं करना है. It is not there in your syllabus, though I'll tell you what does CLA mean, but you don't learn. Chalo, abhi very quickly. First box will show CLA and Hind. Hind which one?
3.
Third.
Hind 3.
Third.
Hind? Yes, Hind 3. Now we'll move on. We are going clockwise. Chalo, we come to this second one. Okay, let's come over here. Now, A, B, C, D आता है. So A, first part is already done. A में हमारा ये part हो गया. This is your B part. So come to this. B. B is your second one, that is BAM H1. Which is the next site that you have to learn? Quickly tell me.
BAM
H1.
This you have to learn. I don't know which original method you will derive, what you will do, but we'll have to learn, children. So this is BAM H1 in the same department over here, in the same section. You have to take one more, that is SAL1. Which is the next two you will take? BAM
SAL1.
H and SAL1. Now tell me of a way to learn these. First section to Hindustan China हो गया. Second section कुछ ऐसा ही सोचो. Give name. Come on, you're such creative children. सोचो कुछ. Nothing. B and S. You can't think of anything about B and S.
Ma'am, biology students.
चलो, very good, excellent. We got a clue. Biology students हमेशा सबसे आगे रहते हैं. They are always number 1, and they are Hindustanis also. So we'll take it as BAM H1 and student number 1. So BS is BAM H1 and SAL1. Clear, everybody? What was the first one? First box?
China, India.
China and India.
China and India.
China and India to clue hai. Keep speaking the names also. It is Class I and Hind 3. Speak.
Class 1 and
Class 1.
11.
Class 1 and Hind 3.
Class 1 and Hindi 3.
Come to the second box. It is BAM H1 and SAL 1. What is this happening? Please mute. BAM H1 and SAL 1. Clear everybody? Chalo. Let's
Yes,
move on
ma'am.
to the third box. This has to be empty. Number 3 is not shubh, not auspicious. Isko khali chhod do. Leave number 3. Come to number 4 now. Over here, we have another one that is PVU 2. You also have physics now. Bio students also have physics. So let's remember like this, something like this. This is PVU 2. Ab 1 to ho gaya, so 1 ke baad we need to come to 2. So this is which one? PVU 2. Tell me the next one after BAM and SAL.
PVU 2.
PVU 2. Tell me
2.
everybody. PVU
PVU 2.
PVU 2.
Yes. Then you leave 2 boxes or 3 boxes. That is all your choice. You can leave again blank. If you want, you can leave 2 or leave 3. But minimum 2 छोड़ना because यहाँ पे 1 छोड़ा था. So either make it 2 or make it 3. That is all your choice. Now we come to this. PVU के बाद we are moving on to, tell me, PST 1. Which is the next one? PST 1. PST 1 के बाद again the same one, यही वाला same, but PVU 2 की जगह we are going to name it as PVU 1. PVU 1 के बाद we know very close to this Indo-China here is this. This is Eco R1. This one. This one is what? This is your Eco R1. Chalo, let me revise very quickly. We have a bada wala circle, we have a chota wala circle. Inside the chota wala circle, we have 4 things. Name the 4 things.
Ampicillin resistant gene, tetracycline resistant
Ori
gene,
site,
and
ROP.
So we are done. We need to have 4 here and we need to name the 8 endonuclease sites outside. The first one was Clavan Hind 2. Oh, sorry, Hind 3. Which was the first box over here? What will you name it as?
Clever and
Class
Class 1